Hello and welcome to the ARS Memorial Lecture interview. Today I'll be talking with the winner of the Sterling B. Hendricks Lecture Award. Welcome to our awardee today, John Clark.
Well, thank you very much. I appreciate this opportunity and certainly thank ARS for kindly selecting me as this year's winner for the Sterling B. Hendricks.
For folks who might not be familiar with it, can you tell us a little bit about DDT and why it's a compound that we're concerned about?
It was probably the first of what we call the super effective insecticides, and it was a really effective insecticides both on agricultural pests and probably more importantly on vectors. It was a very good mosquito-cide. And so DDT just has this long history of events that as a toxicologist you find interesting. At least in the first few years that DDT was used, it was really a remarkable compound because for the first time we actually were controlling pests that were very damaging to the crops that we eat and the vectors that gives us disease. But because of its overuse over time, it started to becoming less and less effective. And for a long, long time, people wouldn't acknowledge the fact that resistance was even occurring because there was some feeling that because it was a synthetic compound that you weren't going to get resistance, which was not true at all. But ultimately what happened is we got a lot of resistance, and about the same time we're getting resistance because of the longevity, the environmental longevity of this compound. We really started seeing some other issues, which was that bird populations, particularly birds of prey and waterfowl and songbirds, started to decrease because of now we know that DDT, or a metabolite of DDT called DDE, was causing eggshell thinning.
So it sounds like DDT was initially almost a miracle product, but then over time, as it was more heavily used, or perhaps, as you said, overused, some downsides started to appear. So when did that start to happen, and how did society react when it became clear there were some negative side effects?
We started using DDT as an agriculture and vector control agent, right after World War II, and people started realizing that there were probably initially some problems, sort of, the mid 50s. And then by 1962, Rachel Carson wrote a book called Silent Springs, which sort of showcased the fact that DDT was one of the agents, probably the major agent that was causing this decline of bird populations worldwide. And at that particular time, people got very, very concerned about what was going on with DDT, and we needed to understand not only the mechanism of resistance, but we also needed to understand what was happening out in the environment. How was it being degraded? Was it being degraded, and what was it being degraded into?
And so what happened? Is DDT still being used today?
Well, very limitedly. It's still used in a few third world countries. Sometimes it's being used illegally, but in some cases it's still a very cheap alternative, and it can work on certain insects. Its major role right now is as a indoor residual spray that they use to control malaria mosquitoes, and that's because the mosquito that controls malaria is called Anopheles, or Anopheles mosquitoes, and they have a behavior where they'll sit on the wall during the day and then come out when lights go down, the sun goes down, they start flying around taking blood meals. But you can go in with DDT and spray the walls of houses, and because it's a very persistent pesticide, it can be there for a couple of years before you have to reapply, and then the mosquitoes just being exposed to it, on the wall where nobody else is really in contact. So indoor residual spraying is still a useful tool in our treatment of malaria.
And is it not being used otherwise because people don't find it effective, or is it actually banned?
Oh, it was banned. We stopped using it for all agricultural purposes about 1972, and then in the Stockholm Convention of 2002, there was a worldwide ban on it.
Okay, so it's still being used, but only in very isolated cases.
Yeah, I mean, it's either being used primarily illegally or it's like I said, registered by the World Health Organization for indoor residual spraying.
Now, I just want to clarify one of the terms you used. You mentioned that it was helpful against vectors. By vectors, do you just mean any kind of organism that transmits disease?
Well, I work on insect vectors, of course, and so when I say it, yes, it's the insects that will transmit disease to human beings. Malaria, dengue fever, yellow fever, those sort of things.
Now, tell us a little bit about what you found when you started to look at Drosophila and the way that it had responded to DDT. What were the questions you were asking?
Some of the early work was done on a fly, house flies from Denmark, actually, where the flies had become so resistant that they would just sit on the barn and not fly away when you sprayed them. And what they found out was that the resistance mechanism was due to mutations in the target site in the voltage sensitive sodium channel. The resistance mechanism was called knockdown resistance because the flies were not knocked down any longer by this neurotoxin. Um, and so we started looking at that, but it's of interest of sort of, we don't know the answer, but in the 91R strain, the KDR mechanism, if it was selected for, it was lost over time during the selection process. So what we found was that there were three major resistance mechanisms. The first one was called a reduced penetration, where you see that the insect, the outer skeleton of an insect called exoskeleton has a waxy layer on it, and this waxy layer is there basically for desiccation purposes. But we found that waxy layer becomes thicker, and also some proteins that make up the cuticle are overexpressed also. So the penetration factor is due to a thickening of the cuticle and an increase in the cuticular hydrocarbons that make up that waxy layer. And because the waxy layer is similar in solubility of DDT, that they're both fat-loving compounds, DDT will just sit on the waxy layer and then not penetrate into the insect. So it's a penetration factor.
So, when you talk about a cuticle, that's a sort of layer of the outer part of the insect that protects it.
Insects don't have bones, and they don't have an internal structure. They have what's called an exocuticle, and the exocuticle is when insects molt, they molt off that old exocuticle and they form a new one. But it's just the outer covering of an insect, and it's a combination made out of polysaccharides and a number of proteins.
So, in the case of the Drosophila that you were researching, one of the ways it was resisting DDT was that outer shell was becoming thicker.
Yeah, and because it had a waxy layer, the waxy layer would absorb DDT, so it wouldn't penetrate. And we found two other mechanisms, and those mechanisms are, one was called metabolism, and again, because DDT is a foreign compound, it's part of the xenobiotic metabolism I talked about, and there's a couple enzymes called cytochrome P450 monooxygenases that will degrade DDT to non-toxic metabolites. They're called dicafol and DDD, and these genes have been upregulated in the 91R strain, so it degrades DDT in a much more efficient manner. So if the DDT is being degraded into non-toxic metabolites, that's what we call metabolic resistance. Yeah, the third mechanism is something called enhanced excretion, and we have a number of ways in which we can excrete compounds. Most of the time we just do that in urine and feces and the air that we breathe out. But in this case, we have a group of transporters that are called ATP binding cassette transporters, or ABC transporters. And these are just efflux proteins that use ATP to excrete DDT out of our body. So obviously that's very effective, because if you're excreting DDT out of your body faster than you're taking it up or metabolizing it, then that becomes this three mechanisms can become synergistic and are probably responsible for the very high level DDT that we see in the 91R strain.
So it wasn't that the fly had developed a single way to resist DDT, it was that it had multiple changes that had taken place in different body systems.
Yes, that's correct, and we see that a lot. I mean, usually when insects start becoming resistant, a lot of times that resistance can be metabolic, but then over time, you'll usually have secondary and tertiary mechanisms that then all starts acting together, which we call synergistic, and that's when you get very high levels of resistance, like 91R, and that's when compounds become ineffective and we have to go and find new compounds to control them.
And why is it important for us to understand the specific mechanisms that organisms develop to become resistant in this way?
Well, the mechanism's important because that shows us exactly what's going on, with how the insect, defends itself away from this toxin. I mean, that's all they're doing. They're evolving their genetics systems to ways in which they can live, by being exposed to this compound that we think is useful, but it's a toxin to the insect. So there's a lot of evolutionary pressure behind using insecticides for a long amount of time and at high concentrations that all organisms will eventually become resistant. And so one of the things that we can get out of this information is that it tells us, what metabolites are being formed. So if we know that, then we can take a look at the structure of DDT or any compound, and we can synthetically alter those parts of the molecule that are being degraded, so it's no longer being degraded by these xenobiotic, metabolizing enzymes. So knowing that is very important. The other thing mechanism allows us to do is that we can go in and we can find what we call biomarkers, and these are things like mutations in genes, like the KDR mutations I was talking about. They can be enhanced enzymes like the cytochrome P450 mechanisms I was talking about, and or they can be the ABC transporters, and then we can either go and sequence and identify the mutations, or we can use enzyme assays to find the enzymes, or we can make antibodies against these transporters, so we can monitor resistance, and that helps us if we go out to a population, we want to think if we have an effective control, you can use these biomarkers, say, well, no, you can't use that compound anymore because it's resistant. So resistance monitoring comes from knowing the mechanism.
Great. All right. So we've covered a lot of territory, and it sounds like there are some pretty exciting applications for this work in agriculture and maybe beyond as well, like public health or really anywhere that we need to control an insect population. So we've gone from looking at DDT and how a certain kind of fly has developed specific ways of protecting itself to talking about some broader applications. So when you think of your work overall and kind of how you hope it will broadly have an influence, what are the positive outcomes that you would ideally like to see?
Well, I think I just kind of mentioned, the first one, which is that that insects, will try to survive, and we call it insecticide resistance, and it's a bad thing for an insect. It's a very good thing. So finding Achilles' heels is probably one of the biggest important things that we can do with the information that we have and coming from this model insect Drosophila. The other thing is that in the case of 91R, we have an insect that's become resistant because it's upregulated. It increased the synthesis of 7 genes and 3 different mechanisms, and we know now that those genes are spread over the 3 major chromosomes in Drosophila, And now that we know what the genes are, we can go in and we can knock these genes down by a number of different things. We can use CRISPR-Cas9, we can use RNAi. It just basically will destroy the transcripts, and so the protein's not created, so you get, susceptible insects back. But we can go back now and knock down these genes one at a time and then in combination, so we can start asking the question, how do these genes interact? And if you in my original talk, when I talked about these resistance mechanisms, I believe penetration was about a 30% reduction in 91R. Metabolism was about a 60% increase in 91R, and excretion was about a fourfold decrease. Well, if you add those together, or even if you multiply those together, that's not even close to a 1,500-fold resistance. And so it's very important that we know that how do these genes interact with each other in the genome, because that's going to help us. Well, what are the most important interactions? And if we disrupt those interactions and we don't get this synergy between them, we can really decrease the amount of resistance that the insect actually has. So knowing something about how genes interact are probably as important as the evolutionary consequences and finding Achilles' heels. But I would think those were the two major things that we found that we certainly thought were interesting.
Well, John, it sounds like there are some really important applications for your work, both in agriculture and beyond. So thank you for taking the time to really help us dig into your research and understand the complex interplay between organisms and some of the chemicals that we've developed to help control them.
Well, thank you very much. I've enjoyed this myself.